Hello everyone. Today I want to jump around a little bit on the center section of this booklet. So the first thing I want to look at is this page about amino acids. We've been discussing how amino acids are what makes up our proteins by making up chains of amino acids called polypeptides. These polypeptide chains are made up of these 20 amino acids that are indicated at the top of this page. 9 of these are considered the essential amino acids. This includes threonine, histidine, tryptophan, methionine, phenylalanine, lysine, valine, leucine, and isoleucine. And those three at the bottom, valine, leucine, and isoleucine, account for 30 to 40% of those essentials that we need. And they cannot be synthesized within your body. So your body can't make these themselves. It must consume them from an external source, usually by eating. On the right, we have the 11 other non-essential amino acids, and this creates the 20 amino acids that we need. So 20 amino acids. And these other 11, you can see the names there. Those ones can be synthesized by your body, usually by consuming protein, breaking it down into its composite parts, and then reassembling them into whatever you need. The eight of the 9 essential amino acids can be found in these products here. This is an example of some of the sources you can get these foods that are non-meat sources. Meat is an appropriate choice as well because meat has lots of protein in it. But of course, if you eat lots of nuts, lots of eggs, lots of chickpeas or seaweed, seaweed apparently is a superfood. It has all of the things that you need. So eat a lot of seaweed, eat a lot of sushi, and you should have lots of amino acids to go around. All right, I'm going to roll up a little bit, and we're going to go and look at this page here, where we can see how those see some coding practice. So this directly builds on our example that we did, or builds on what we talked about in our transcription process. So in transcription and translation, we take a DNA code and create an RNA code. So this is going to be our transcription. So you can see here that I took a template strand that I was given, and assuming that this was all exons, and no, this is only exons. Oh, I just found an error. So this is a strand that is all exons, so it's all going to be carried through to our final polypeptide. We take the sequence, we match each of the bases with a complementary RNA base. So here I took T and I matched it with A matched with U, C matched with G, and carried that forward all the way across. Just about like that. There we go. And carry that all the way across until I completed the sequence. I don't quite know where it's going to stop. Remember that the mRNA will be written out. It will start from wherever the transcription complex forms, and it will read out and create that mRNA all the way to the end until it hits that termination sequence. So though the mRNA might still be longer than the amino acid chain that you're going to create, the amino acid chain, on the other hand, will only start at the AUG sequence. So luckily, in this example, the AUG is right at the front of the strand, but it could be anywhere in the strand. That AUG will call down a methionine, and then I will use this codon table to find out all of the other ones. So AAA, I go to the codon table, I find AAA right here, and that is a lysine. So the lysine goes up top. GGG, I gotta go down a little bit more farther. GGG we can see here, and that is a glycine. So that goes next. CGC, etc. That's an arginine. And we go along until we get to this sequence, UAA, and we find UAA is a stop codon. So we just list stop. That is not an amino acid. It is just the end of translation. And so this process here is the translation process. And so at the end of the day, we have 1, 2, 3, 4, 5, 6, 7. We have 7 amino acid polypeptide. All right, so we can do the same thing in the next example, but in the next example, I took a similar sequence of DNA and I converted it into a non-template strand or a sense strand. So this sense strand of DNA has to be converted into the template strand first. So this is just complementary base pairing. So the A with a T, T with an A, G with a C, TA, 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 etc. Now I have my template strand. Now I can perform my transcription. I'm just going to steal this one. So now I can perform my transcription, and then I can perform my translation on that transcription. And so then the translation will be performed on that transcription. This time, I am still going to be looking for that AUG start sequence. And notice that because I started with a template strand, even though it was the same strand that I had before, I can see that because I was starting from a different complementary strand, I ended up with a stop codon at a much earlier position. And so in this example, I only formed 1, 2, 3, 4, 5. I formed a 5 amino acid polypeptide from the 6 codons that I translated, because that last codon was just a stop. All right. Another nice thing to look at is when you have a sense strand, is you can use the sense strand to help you work out your DNA, your mRNA sequence. So notice here GGG, GGG, GCG, CCG, CCC, CCC. So the sense strand has the same coding as the mRNA because both strands are complementary to the template strand. And so that's how we do that part. So hopefully that helps you out, and I will be posting some questions that will help you have you apply this codon table to some other problems. So keep this codon table handy. It will come in to play on a variety of little assignments that are coming. And the other page I would like to talk about is the one that comes right before this, which is about protecting this code. So now I'm hoping that you'll have some appreciation for how the DNA sequence controls the RNA sequence, and that RNA sequence controls the amino acid sequence, and those all together create the protein which is going to control the function in the cell. And all of the traits of the cell will come from that part. So that's that central dogma, the central idea of modern biology, that DNA controls RNA, RNA controls amino acids. So if there is something that's going to change the DNA sequence here, we're talking about the order of the bases, then there are at least a couple of factors that we should consider that protect the sequence from changing. The first factor is that purines can only match with pyrimidines in the double-stranded DNA. So this limits the types of mutations that can happen. So mutations do happen in the DNA strand. The DNA strand can be mutated by errors during replication. It can be mutated during errors in transcription or translation. The wrong base gets into the wrong place and causes that change. But there are some limitations here. Mutations typically only a purine replaced with a purine will be undetected will remain undetected and will be able to be transferred to the cell, and the cell will be able to continue to live with that change. So that means that cytosine and thymine being replaced with one another, that has a much higher chance of happening than a cytosine replaced with an adenine. That's much less likely. So let's look at that. Let's keep that in our mind. So cytosine and thymine, that's a possible mutation. Look at this first example here of phenylalanine and the codons that are associated with it. What do we notice about the codon? Well, there are 2 options for phenylalanine, and in that option, one option ends in a U, one option ends in a C. as in they both end in a purine. And for leucine, they both end in a pyrimidine. So in a way, the codons have already taken into account that there is a possibility that purines could be replaced with purines. Purines could be replaced with purines, or pyrimidines could be replaced with pyrimidines. And so these are actually already, these factors have already been taken into account in the way that the codon table functions. And so that's kind of an amazing ability of our DNA. Over these billions of years of its development, of its evolution, it incorporated the likelihood of transformation, the likelihood of mutation has been incorporated into the codon table itself and helps prevent the codon table from actually being affected by these mutations. So even if there was a mutation that caused a uracil in this position to be replaced with a cytosine, we would have no impact on the protein itself. That's kind of unbelievable. I think that's kind of incredible. So that kind of fits into my, my third point here, that out of the 22 amino acids, since there are only 22 amino acids, in fact, we've actually been discussing only 20 amino acids, so let's just fix that. 20 amino acids, we have 64 codes. And so by having more codes than amino acids, some mutations will not change the amino acid sequence. In fact, the most common, many of the most common mutations have no impact, such as a uracil replacing a cytosine. In what was that one? It was UUU and UUC. UUC and UUU both call up phenylalanine. Why did I have to pick the one that has the longest name possible? I think it's phenylalanine. That's my problem here. Phenylalanine. Now, the other factor that we've discussed previously was that I believe in our data, in our earlier thing, we said 1.5. So only about 1.5% of the DNA actually forms exons. So that means that there's a whole bunch of DNA there that can accept mutations without impacting protein. So this implies that a change to the DNA has only a 1.5% chance of affecting the protein coding sections. And so once you combine that effect onto the other protective parts, then you actually have a very minimum chance of changing the DNA. And that's one of the ways that the mutation rate can be slowed. The mutation rate is held down. It's not high, it's not as high as it possibly could be if there weren't these other factors. The final one that's not probably in your notes right now, but you can add this, is that polymerase 1 and 3 are always around in the nucleus, and those polymerases are always going along the DNA sequence, and they're actually fixing some of the mismatched pairs, and they will repair these mutations. This has actually been, there are other DNA repair processes. There are also additional DNA repair processes that can correct for mutations. And this has actually been a problem in cases where they've been trying to purposely change the DNA through things like genetic modification. So in genetic modification, they're trying to change the DNA, but they are actually having difficulty getting those changes to stay through multiple generations of the cell. There are processes in the DNA and in the nucleus that can repair those changes that they're purposely making through a variety of techniques, and some that are still being discovered today. All right, I'm sure I'll have more to say about this section in a future video, especially as we enter into our next unit on our next section of the unit, which is all about mutation, and then we'll close out by discussing genetic modification and some of the basics and ideas about how we are modifying our organisms to improve our future, and how it might be the sole solution to all of our future problems. So hopefully those will be interesting to you, and I hope to talk to you about those soon. Have yourselves a great day.